for 8.2, you need to understand the pieces, the structure in DNA. And these monomers are called nucleotides. The nucleotides have 3 parts. Do you guys see this? Starting with the nitrogenous base. There's 4 different nitrogenous bases. So there's 4 different nucleotides. Then what's this, everybody? That's like a pentagon. It's a sugar. And what's the name of the sugar? I tricked you. Deoxyribose. See the os at the end? Okay. Glucose, fructose, all those are sugars. It's a sugar. This is where DNA gets its name. DNA, deoxyribo, and then it goes nucleic acid. Okay? Now, so DNA, deoxyribose nucleic acid. Then your phosphate group. These guys make up the backbone. The bases make up like the rungs to the ladder. Oh, no, I had a little thing. I can't find it. Okay, on to your power notes. You need to write down. Thymine, I put big T next to it. Cytosine, big C. Get these 4 nucleotides. So your 4 nucleotides. Thymine always bonds to adenine. Right here. Hurry. Whatever one you're in. For real. Adenine and guanine. C always bonds to G. T always bonds to A. It's how they fit together. You can see that these ones over here have 2 separate rings. The ones over here have single rings. And a single ring fits with a double ring. And they only pair up a certain way. That's very important that you guys realize that. Okay, under base pair rules, do you guys see it? Under base pair rules, copy it down. A pairs with T. C pairs with G. I memorized this with the word at. There it is, A and T together. Some people like to memorize it with the capital C, kind of looks like a capital G. Mr. Hager remembers it with C in G, G and C. G and C is a health food store you guys might know of. So he memorized it that way. It doesn't matter. Whatever works for you, you have to have the pair bonding memorized. Okay, now where are we at? Structure. It's A to T and C to G. You guys need to join me. Moving on here. Okay, this is under bonding, number 1 and number 2. Quickly. Okay, so the solid bond lines on here are covalent bonds. Those are stronger bonds between the sugar and the phosphate, between the sugar and the nitrogen-containing base. These dotted lines, what kind of bonds are these that are connecting the bases? Hydrogen. Notice, are A and T strongly bonded as G and C? No. 2 bonds here, 3 bonds here. Now that's kind of interesting. We have realized that organisms that live in harsher environments have more G's and C's in their DNA than A's and T's. Like, for example, bacteria in hot springs. High temperatures are going to make the DNA more fragile and come apart. So it's better to have more C's and G's. Okay, moving on. Now, over on the right side, you must know the shape of DNA. It's known as the double helix because double, it has two strands, two sides, two strands, and it's doing a helix, which is a spiral. Now, what about the structure of the backbone? The structure of the backbone is alternating between a sugar, which is deoxyribose, that's the red on my model, and a little phosphate group, back and forth. That's the backbone. Now, Watson and Crick get the credit for discovering the shape of DNA. However, they did have lots of help. Okay, before we go on, draw a line. So from your sugar right here where you wrote it, point at the sugar on the model, which is the pentagon, and then the little phosphate. Now, see what I mean? Does this drawing diagram do a better job? Not really. They still have a pretty big phosphate to sugar, don't they? It's just to make it fit on the paper. The phosphate group's the circle, so draw a line to the circle, and then draw a line from the sugar to the pentagon. That's your sugar part. Now here are Watson and Crick. One was from England, worked at a university there. One was from America. Okay, but they mostly did their work in England. They made these big models to try and figure out the placement for the shape of DNA. Now, they got a lot of help, and in an unethical way, very controversial. This is back in the 50s. So, and Rosalind Franklin has been since passed away. She died a while ago. So there's some legends with the exact precision of the story. However, we do know that she took these photographs of different molecules. They're called crystallography. And what she was doing was she was making crystals out of molecules. She was basically drying molecules out, then hitting them with radiation and making X-ray photographs. And in that way, she was getting the shape. She was getting shadow imprints of the molecules. And she was also trying to determine the shape of DNA. And she got this famous photo. And if you look at it closely, if you think of going down the side of DNA, you would see spirals. This would be making circles. And when it's pretty abstract for me, but when you've been taking photos of different geometric shapes, you start to recognize the patterns really easily. And this is making an imprint of a spiral. So this was amazing. She noted this. Now, where the story gets a little wacky is that one of the graduate students that worked with one of her he wasn't a graduate student, he was a younger scientist. His name's Maurice Wilkins. And anyway, they did some sharing and talking, which is normally happens in science. But you're all supposed to always give credit, and you're supposed to get permission, right, before you share, and you want to publish and all that. Well, Wilkins shared this photograph with Watson and Crick, and it was the aha moment. They were able to put their pieces together in spiral shape. They had not thought of a spiral, and it fit together perfectly. So the real problem came when they didn't give any credit. They didn't talk about the photograph. They just took the Nobel Prize, took all the fame, and didn't tell the truth for some time. Then they finally told the truth. Crick was more honest. Crick is still alive. Watson has since passed away. Watson was kind of a brat about it till the end and had said awful things about Rosalind, and it got really weird. There's some cool, interesting documentaries you guys can watch on it. There's another person, Chargaff. I can't remember his first name. He was also working on this. His discovery also was shared with Rosalind and with Watson and Crick, and this was also helpful. And this should make some sense to you guys now. So he was measuring the amounts of DNA. How many A's are there? How many T's are there? How many C's and G's? Well, the amount of A's always match the amount of T's. Same amount of A's to T's. Same amount of C's to G's. Why would that be? They're bonded together, right? The letters are they name the molecules first. They name them adenine. Chemistry is pretty cool, and you guys take it next year if you do. The naming follows strict guidelines. The name is revealing if you're a chemist. You know what's included in them. So then it was just their abbreviation by letter. So anyway, does this make sense, you guys? Chargaff is like, hey, there's always the same A amount as T. Well, if they're bonded together, does that explain why there's always the same amount? So this also, so here's 3 or 4 different scientists doing this work, all coming up with the same thing. It's just kind of too bad that it wasn't done more ethically. Okay. Next one, we'll go quicker, so you guys see the next section. Oh boy. Okay, try again. Okay, 8.3, you guys. Now, the structure of DNA makes it copy itself almost perfectly every time and copy itself very easily, just because of the shape. This is called replication. Replication copies the genetic information of DNA of a cell during the cell cycle. Now back up. Do you guys remember the cell cycle, the pie-shaped thing-a-majigger? Remember I told you DNA copies itself in one of those phases? Do you guys remember what phase that was? Not in mitosis. Before mitosis in? Before gap 2. It was in interphase. And it was called the S phase. So, do you guys remember this? Look at this. There was nothing to write down from that. So, listen up. Okay, so with a single strand of DNA serves as the template. I'll show you in a minute. But we're talking about this S phase right now. This is where DNA is synthesized, and we call the process replication. Okay, now, I am not going to tell you these. I want you to process and think. Look carefully. The diagrams are slightly different. This enzyme, DNA polymerase, it is attaching the nucleotides by what pair, the pair bonding. If there's an A on that side of the DNA, it's going to put a, if there's an A, it's going to put a T. Okay, so do you guys see your oval? DNA polymerase. Now, what's really cool, look carefully, the new side that is being built, it's being built off the old side when the DNA was opened up. So the new strand, I think that's number 4, correct, you guys? Now, number 1, it was pointing to the new strand and the null strand, so they're just talking about the sugar phosphate backbone in general. Okay, now this is called DNA replication. Okay, copy down number 1 and 2, and then I'll explain. Okay, people, focus so you get this right now. So, for the DNA to copy itself, what it has to do is it has to unzip. It's just those hydrogen bonds. The hydrogen bonds are weak. It opens up in this little bubble shape. We got on one side it happening in one direction, and on the other side it happening the opposite direction. So the enzymes, gentlemen, is this going to work over here? The enzymes unzip the double helix, so that's that big floaty guy, in 2 different directions, and then they also attach the nucleotides, And this is, you need to make sure you're eating properly and getting everything you need. Because if you're missing, some nutrients, then we don't have the nucleotides or the amino acids for your proteins. You guys need to be feeding yourself, otherwise your body, your cells can't make what it needs. Okay, so that is number 1 and 2. Let's go on the next one to number 3. Okay, so the last step is the DNA polymerase enzyme bonds the nucleotides together. So we have to get the backbone also bonded together to form the double helix, these covalent bonds, and we get 2 strands of DNA again. Now, eyes up here. Look at this closely, you guys. This is called semi-conservative replication. It's not like I showed you this model and gave you all the pieces and said, hey, go build that, right? It's not like that. It's pretty cool. It unzips. If you know one side, can you build the other side? Yeah, because the A to T, C to G. And now, so it just unzips, and you're getting both of the strands being rebuilt individually, and they've tried to color code it. So this is also interesting because normally when I say, hey, go copy something, right? I give you one thing, hey, go copy this. You come back with something brand new. Here's the old one, there's the new one. Not in this case, which if you think about it is pretty cool because like you probably have little fragments of pieces of your father and your mother's DNA that directly came from their sperm and egg. That was in the cell from them. And then so forth and so forth. So you could have some prehistoric little chunk of DNA that's now in one of your DNA strands. So it's kind of cool. That's called semi-conservative replication. The old one doesn't get broken down. So you get just like this. And think how exact it is. So once again called semi-conservative because the old strand, half of it, just part of the DNA is old, so it's been conserved. I'm not going to worry about it too much. Okay, just the last slide, nothing to write, but I want you guys to realize, look up here people, that these bubbles, these openings will happen all along the big long piece of DNA, so it gets copied really fast. Now, bacteria doesn't do it that way. Bacteria has one circular chromosome, one circular shape. It just goes from one end to the other. All right, you guys made it.